Ketogenic diet and retina, protecting eye nerves through metabolism. Glaucoma is an eye disease where pressure or other factors cause progressive damage to the retinal nerve cells, retinal ganglion cells, RGCs, and their fibers, leading to vision loss. Traditionally, treatment focuses on lowering eye pressure, intraocular pressure, IOP. Recently, researchers have explored whether changing body metabolism, for example, with a ketogenic diet or ketone supplements, could help protect RGCs. A ketogenic diet is very low in carbohydrates and high in fats. In response, the body burns fat and produces ketone bodies, like Beta-Hydroxybutyrate or BHB, as a fuel. Ketones can serve as an alternative energy source for the brain and eyes. Emerging evidence suggests these metabolic changes can boost cell energy use, quiet harmful overactivity, excitotoxicity, and even alter gene activity in ways that may shield RGCs from damage. In animal studies, ketone treatments have improved RGC survival and function. In other model systems, BHB shows broad anti-inflammatory and longevity effects. In this article, we explain these findings in plain terms and discuss what they mean for glaucoma patients, especially those who are older or have other health issues. Fueling mitochondria, energy efficiency, and retinal health. The retina, especially RGCs, is a highly active tissue that needs a lot of energy to work. This energy comes from tiny structures in cells called mitochondria. If mitochondria work better, nerve cells are healthier. Ketones are a special fuel for mitochondria. They can be turned into energy efficiently, sometimes even more cleanly than sugar. A number of studies have shown that ketogenic metabolism boosts mitochondrial function. For example, one study in mice used a glaucoma model and found that a ketogenic diet promoted both mitochondrial biogenesis, making new mitochondria, and mitophagy, recycling damaged mitochondria, in RGCs. In that glaucoma model, mice eating a high-fat, low-carb diet kept more RGCs alive than control mice. The investigators noted increased mitochondrial markers and better energy balance in those cells. In simpler terms, the ketogenic diet gave the eye nerves a metabolic upgrade, more and healthier mitochondria that could meet energy needs under stress. Animal research also links ketosis to better antioxidant defenses, fighting cell damage. For example, a scientific review points out that ketogenic metabolism can lower production of harmful reactive oxygen species and boost cell protective pathways. In experimental glaucoma, an inherited model in DBA2J mice on a ketogenic diet showed healthier mitochondria and more antioxidant response compared to controls. These changes were accompanied by better RGC survival. This suggests that providing ketones, either through diet or supplements, may make retinal neurons more energy efficient and resistant to stress. Calming excitotoxicity, dampening overactive nerve signals. Another stress factor for neurons is excitotoxicity. This happens when too much glutamate, a common nerve messenger, overexcites cells and leads to injury. In glaucoma and other neurodegenerative diseases, excitotoxicity can kill retinal cells. Laboratory studies show ketones can attenuate this effect. In one rat study, scientists gave animals a toxic dose of NMDA, a glutamate-like chemical, to kill RGCs, mimicking excitotoxic damage. Rats that received injections of beta-hydroxybutyrate, BHB, or acetoacetate had significantly less nerve cell death than untreated rats. In other words, ketone bodies protected RGCs from a glutamate-related assault. The researchers found that BHB helped preserve levels of protective molecules like kynurenic acid, which naturally block excitotoxic signals. This suggests that ketones can act like a brake on harmful nerve overactivation. The exact mechanisms still need more research, but the effect is consistent with the known anti-seizure benefits of ketogenic diets in epilepsy, which also involve reducing excitotoxic activity in the brain. For glaucoma, dampening excitotoxicity could mean nerve cells are less likely to be killed by the constant stress of high eye pressure or other insults. Thus, ketogenic therapy may protect RGCs by both boosting their energy and keeping their electrical activity in a safer range. Epigenetic effects. BHB as a gene regulator. Beyond immediate fuel, ketones have surprising epigenetic effects. They change how genes are expressed. The key player here is BHB. Beta-hydroxybutyrate can influence the modifications like acetylation of histone proteins that package DNA, as well as other gene-regulating proteins. These changes can turn on long-term resilience programs in cells. For example, BHB is known to inhibit certain histone deacetylase enzymes, HDX, which normally act to silence genes. By blocking HDX, BHB tends to loosen up chromatin and allow protective genes to be more active. In brain and cellular studies, BHB's HDAC inhibition has been linked to anti-inflammatory and antioxidant gene expression. In fact, research cited in Science showed that BHB treatment raised the acetylation of histones in the brain and activated key stress response factors. In retinal cells specifically, inhibiting HDACs has been shown to preserve RGC structure. While direct studies of BHB's epigenetic effects on the retina are still emerging, the known action of BHB implies it could turn on genes that protect nerves. Animal experiments reinforce that this epigenetic switch matters for longevity. In the roundworm C. elegans, giving BHB extended the worm's lifespan. The mechanism required the worm's equivalent of FOXO-DAP16 and NRF2, SKN1, key longevity regulators, and also depended on BHB's HDAC inhibition. In other words, BHB extended life by activating well-known longevity pathways, and it could not do that without affecting histone acetylation. This suggests that in higher animals too, BHB might promote a protected gene expression profile in nerve cells, helping them survive longer under stress. Anti-inflammatory role, calming chronic damage. Chronic inflammation is another problem in aging and glaucoma. Damaged tissues often raise inflammatory signals that can harm neurons. Here again, BHB has useful actions. One major target is the NLRP3 inflammasome, a protein complex that drives inflammation. Studies show BHB blocks the NLRP3 inflammasome, thereby lowering pro-inflammatory cytokines like IL-1 beta and IL-18. In fact, Umet et al. 2015 in Nature Medicine found that BHB stopped NLRP3-driven inflammation in several disease models. Specific to the eye, a recent study in diabetic mice looked at this explicitly. The researchers injected BHB and found it dramatically reduced markers of inflammasome activity in the retina. After BHB treatment, diabetic mice had about half the levels of retinal NLRP3, ASC, and caspase-1 inflammasome components, and much lower IL-1 beta and IL-18 compared to untreated diabetic mice. In short, the ketones stop the chain of inflammation signals that normally builds up in diabetes. This is promising because many glaucoma patients also have diabetes or other conditions that cause chronic eye inflammation. By acting on HCA2 receptors and inflammasomes, BHB could help quiet retinal stress. Additionally, BHB can bind to G protein receptors, like HCA2, that have anti-inflammatory effects in tissues. It also has been reported to scavenge free radicals and support antioxidant pathways. All these signals together mean ketones teach cells to switch from an angry, injured mode to a protected, healing mode. In model organisms, this shift is linked to health span. For example, rodents on cycles of ketosis showed less brain inflammaging and better cognitive performance in old age. While more human studies are needed, the anti-inflammatory properties of BHB are now well documented. Ketones and longevity, lessons from lab models. Laboratory studies of aging hint that ketosis could be part of a pro-longevity regimen. In simple organisms, supplementing with BHB can extend life. C. elegans given Beta-Hydroxybutyrate lived significantly longer and showed better stress resistance. Their longevity increase required the same FOXO and AMPK pathways known from calorie restriction and needed BHB's HDAC effects. This supports the idea that ketones mimic some benefits of fasting, like activating housekeeping genes. In mice, the data is mixed but intriguing. One study put middle-aged mice on a cyclic ketogenic diet, alternating ketogenic weeks, and found that fewer mice died in midlife, and their memory stayed sharper into old age, compared to controls. The ketogenic mice showed gene expression changes in common with fasting or high-fat feeding, including activation of PPRA targets involved in fat metabolism. Importantly, memory tests like object recognition were much better in the ketogenic group, even at toward 2 years old. This suggests ketosis helped preserve cognitive function with age. No such study exists yet in humans for glaucoma, but it hints at possible brain benefits. Finally, a general review noted that ketones and related short-chain fatty acids repeatedly show positive effects on health and lifespan across species. However, it also warns that long-term use needs study. Indeed, a very recent mouse study found that lifelong, unrelenting ketogenic diet led to high blood lipids and liver issues in mice, along with drop in insulin secretion. That means any recommendation must balance benefits with risks, especially over many years. Clinical considerations: who should try it and how? Glaucoma patients are often older and can have other health issues like diabetes, high blood pressure, or kidney concerns. Any change to a strict keto diet needs careful thought. Here are some key points. Diabetic patients. Type 1 diabetics must be extremely cautious. Lack of insulin plus a keto diet could trigger diabetic ketoacidosis, a dangerous condition. Type 2 diabetics also need monitoring. However, short-term keto under medical supervision can improve blood sugar control in some patients. Anyone with diabetes should only attempt ketogenic diets with a doctor's guidance and frequent blood tests. Heart and blood lipids. Because ketogenic diets are high in fat, some studies report rises in LDL bad cholesterol. Others show improved markers, like lower triglycerides in overweight people 41. Still, a patient with known atherosclerosis or very high LDL should consult a cardiologist. It would be wise to check cholesterol regularly. Some experts advise focusing on healthy fats, olive oil, nuts, rather than saturated fats if trying low-carb eating. Kidney issues. High protein or ketogenic diets can change mineral balance. One well-known effect is an increased risk of kidney stones. For example, a child on a strict keto diet developed painful kidney stones within months. For glaucoma patients with any kidney disease or history of stones, caution is needed. Good hydration and dietary advice, such as citrate supplements, might help, but this should be managed by a doctor or dietitian. Nutritional balance. Ketogenic diets can be low in fiber, vitamins, or minerals if not planned properly. Long-term ketogenic eating should be accompanied by a vitamin-mineral supplement and possibly a registered dietitian's guidance. Goals like controlled carbohydrates, as in a Mediterranean or modified Atkins diet, might be safer alternatives. Medication interactions. Fasting-like states can alter how some drugs work. For instance, glaucoma patients on eye drops or pills to lower pressure should know that severe dietary changes might affect blood pressure or fluid balance. Patients should not change their glaucoma treatment without consulting their eye doctor. In sum, while ketogenic signals show promise in lab studies, most glaucoma patients shouldn't go keto on their own. A doctor or clinic should supervise any such intervention, ideally with input from nutritionists and primary care. Those with heart disease or kidney problems should especially get clearance. The no free lunch rule applies. If one tries to increase ketones, the potential benefits must be weighed against known metabolic changes. Pilot trials, testing ketones in glaucoma patients. To move forward, carefully designed research is needed. Instead of everyone immediately adopting a keto diet, small pilot clinical trials could test feasibility and outcomes. A useful study might enroll glaucoma patients with and without issues like diabetes and give a monitored ketogenic diet or ketone supplement for a few months. Key measures would include metabolic phenotyping, regular blood tests to check ketone levels, glucose, cholesterol, triglycerides, and kidney liver function. Insulin sensitivity tests could show if the diet is helping or harming metabolic health. This ensures safety. Retinal imaging. Techniques like OCT, optical coherence tomography, can measure nerve fiber layer thickness, a marker of RGC health. Researchers could see if these structural measures stabilize or decline more slowly on keto versus control. Functional tests like visual fields, perimetry, or a contrast sensitivity test could also be recorded. Neuroprotection markers. Special retinal scans can detect metabolic stress, for example, retinal oximetry. Researchers might also measure cerebrospinal or blood biomarkers of neuroprotection, e.g. BDNF levels, inflammatory markers like IL-1 beta. Cognitive aging measures. Since BHB affects the brain too, simple cognitive tests, memory, attention, could be included. If keto really boosts brain health in older age, patients might perform better on standardized memory tests or show improved mood, fatigue scores. Before and after comparisons in control groups. Perhaps an exercise and healthy diet group would help isolate effects. Even if a pilot is too small to prove glaucoma benefit, it would show who can tolerate the diet and whether retinal measures change in a positive direction. Ideally, these trials would be double-blinded, if using a ketone supplement, like ketone esters or salts versus placebo, to control for diet differences. To summarize, basic science suggests ketones from diet or pills could help retinal neurons by improving their energy use, damping harmful signals, and turning on protective genes. BHB in particular is a natural anti-inflammatory signal that activates longevity pathways. These effects have been seen in animal glaucoma models and aging studies. However, ketogenic approaches are not risk-free and require medical oversight. In glaucoma patients with multiple health issues, any new diet should be matched to their overall condition. Carefully conducted pilot studies with metabolic and imaging measurements, plus memory cognition tests, could clarify whether advancing ketone metabolism safely slows vision loss or cognitive aging. As of now, patients should not make drastic dietary changes without professional guidance. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.